This conference will now be recorded. This conference will now be recorded.
Good morning, students. I'm Dr. Divya from TeenEinstein. This is 11th chapter, Biotechnology Principles and Processes, Part 3. So we know that all the processes like new inventions, all new inventions in life science, it's for increasing the production level, food production level, then medicinal production, everything we want to increase. So for that purpose, we are applying new biotechnological processes, right? So to find out a better food source, for that purpose, we are using so many technologies, isn't it? So biotechnology means that is a kind of modern biology. In that we are applying or we are thinking about the applications of biology. So that made a lot of differences or that lot of changes in our daily life and the products, so many products which we are using, that and all. As a result of this biotechnological application we got, right? So in part 1 and part 2, we had a discussion about few techniques and the basic principles behind biotechnological processes, right? We had discussed about tools of rDNA technology and recombinant DNA technology, and which are all tools, what and all we are using for this rDNA technology. So which are the main tools of what and all we are using for rDNA technology? We need a restriction enzyme, then polymerase enzyme, ligases, all these three. First one, second one, and third one, all these are enzymes. Restriction enzyme, that is called as molecular scissors, then polymerase enzyme, ligases. Ligases is called as molecular glue. Then vectors, we need vector. For what purpose? To introduce this gene of interest into the host. Then host organism, we need a selected host organism because all vectors and all gene of interest that can't multiply in all host organisms. So we need a selected host organism also. So these are the important tools of rDNA technology. And in part 2, we were discussing about restriction enzyme. See, mainly there are two types of restriction enzymes, restriction exonuclease and restriction endonuclease. In this restriction endonuclease, that is called as molecular scissors. Why restriction enzyme, this endonuclease, why it is called as molecular scissors? That is helping to make cuts. Okay, so that is helping to cut the double-stranded DNA. So for that purpose, it is called as, because of that property, it is called as molecular glue, restriction endonuclease. Okay, this biotechnology, this part is very important for your NEET exam because so many multiple choice questions and application type questions, most of the questions will come from this part. Okay, so note down. In these two, like exonucleases and endonucleases, endonuclease is making cuts in double-stranded DNA. You know, DNA is double-stranded. Single-stranded DNA is also there. But here you are learning about double-stranded DNA, right? Double-stranded DNA is helping for copying. Okay, that's why double-stranded DNA is called as semi-conservative. Half of the portion is conserved. Okay, when it is going to zygotic level and all, half of the DNA, it is conserved. Then, so exonuclease and endonuclease, these are the main types of restriction nucleases. This restriction enzyme, it is belonging to a large group of enzyme which is called as nucleases. And there are two types of restriction enzymes, restriction exonuclease and restriction endonuclease. So in these two, restriction endonuclease, that is restriction endonucleases is called as molecular scissors, that is making cuts in specific positions, right? So which are those positions? Actually, restriction endonuclease, that is identifying the palindromic sequence. What do you mean by palindromic sequence? From both sides, we can read. We'll get the same sequence. From both sides, we are reading also. We'll get the same sequence. Okay. So that this restriction endonuclease, it is getting attached to this palindromic sequence, not exactly at the position, little away from that sequence. Okay. And after attaching to that sequence, what it will do, it will make small cuts. It will make cuts, that enzyme makes cut in both the strands. Both of the DNA strands will get the same type of cut at the same side. Okay, the DNA is double stranded. So both the strands will get the cut at same place. Okay, in one strand it will make in one position and in another strand it will make little away from that position. It's not like that. Exactly on same place on both strands it will make cut. Which one? Which enzyme? Restriction endonuclease enzyme. Okay, so at the same site it is making cut. And then what it will do? Both in the vector DNA and in the foreign DNA. See, we need to attach the foreign DNA. What do you mean by foreign DNA? Foreign DNA means the DNA strand with the gene of interest, the DNA fragment which is having the gene of interest. Which gene we want to introduce or which gene we want to multiply, that gene of interest will be there in that fragment of DNA. Okay, so that fragment we need to introduce into a host DNA. So while attaching this foreign DNA with the vector DNA, that should have same type of ends, right? To attach both, yeah, exactly, to attach both, that need same type of ends. So what it will do in the vector DNA also, like this, the same restriction endonuclease will make. Okay. So there a sticky end will form, right? So the vector DNA and the host DNA, that both will join at the sticky ends and that forms a recombinant DNA. Okay. So up to that, we have learned in the last class. In part 2, we were discussing about this recombinant DNA. So that's how a recombinant DNA is forming. It is getting attached at the sticky ends. So both the vector DNA and the foreign DNA, both will be having the sticky ends and it will get attached. Okay. Next, then we know that we need to cut the vector and we need to cut the source of DNA with the same restriction enzyme. For what? To create the recombinant DNA, right? There is a recombination. From the normal combination, we are making a recombination. That's why it is called as recombinant DNA. So there comes the importance of cutting the DNA into fragments, right? We need fragments of DNA. Then only we can attach the fragment of DNA with the vector DNA. Got it? So how to make this DNA into fragments? That is the next question. The procedure of making DNA into fragments. How to make these DNA fragments? It's not a simple process like cutting a long thread with the help of scissors or something. It's not like that. See, we can't cut the DNA. DNA is a big strand, right? If you are uncoiling the DNA, we'll get 2 strands. We can't cut it easily. So with the help of restriction endonuclease, we know that how to make cut in DNA strand or how to make DNA fragments with the help of restriction endonucleases. So restriction endonucleases, what it will do, that makes the DNA into fragments. Okay. Then after cutting, we can separate these fragments. So for attaching to the vector, restriction enzyme is making cut to the DNA fragments, right? So after that, we need to separate and take these fragments. So the technique by which DNA fragments are getting separated, that technique is called as electrophoresis. So what is electrophoresis? The technique which is used to separate the DNA fragments. After cutting, we need to separate the DNA fragments which is having the gene of interest. So we need to get it separated. So how to separate that? For separating, we are using a technique. That technique is called as electrophoresis. So you know that DNA is negatively charged. What is the charge of DNA? Negative charge. DNA is negatively charged. So DNA fragments, they are getting separated by forcing them to come towards the opposite charge. So how to make this DNA in a separate way, or how to make the DNA fragments? How to get your gene of interest in a DNA fragment, or how to make that fragments of DNA, how to separate that fragments of DNA. The DNA fragments, they are negatively charged. So we are separating them by forcing them to move towards the opposite charge. So we have learned about opposite charges that will attract, right? A negative charge that will move towards positive charge, right? So same charges will repel. So here these DNA molecules are getting separated. How we are separating? By forcing them to move towards the opposite charge. Or DNA is negatively charged, that will move towards anode. Anode is having positive charge. Under electric field, will make the movement of DNA. DNA which is negatively charged, and it will move towards the positive charge. Okay. So for this, we are using a medium. Which medium we are? That medium that is called as medium or matrix. Of which one we are using? Agarose. Agarose, that is a matrix which we are using commonly for this electrophoresis. Okay, for preparing the gel. So actually, in agarose gel electrophoresis, here for separating the DNA, we are using agarose gel electrophoresis. The DNA fragments, they'll get separated according to their size. Here you can see the gel. This is the agarose gel or typical agarose gel electrophoresis. So here in these wells, we are loading DNA, and this DNA, since isolated DNA will load inside this, and when we are applying the electric field, what will happen from this DNA which is negatively charged, that will move towards the positive charge. Okay, so it will move. That movement will be according to their size. Got it? That means the fragment is small. If the fragment is very small, that DNA fragment can move so much distance. Okay. So if it is, see here you can see the size is more, means it won't move farther. Okay. If it is a small sized fragment, DNA fragment, that can move. The distance, the movement and the distance, it depends upon the size of the DNA fragment. Got it? So cutting of DNA by restriction endonuclease, that makes the fragments of DNA. So these fragments that we can separate by using which technique? The technique is called as gel electrophoresis. Okay. So the DNA fragments which are negatively charged, that will move towards the positive charge. And that's how we are separating the DNA fragments. And for this, a matrix which we are using, the commonly used matrix is agarose. What is agarose? That is a polymer, a natural polymer that is extracted from seaweeds. Okay. Then the DNA fragments, according to their size, that will move through this matrix. That's how the fragments are getting separated. Got it? Next, I'll explain the procedure of agarose gel electrophoresis. Here you can see so many pictures you can see. I'll explain one by one. You have to remember this sequence because This part is important for NEET, especially for NEET exam. This is important. Okay. So Agarose is very important. That is a very important component of this gel electrophoresis. Agarose, that is, it is a natural polymer. It is getting extracted from seaweeds. Okay. Then what is actually electrophoresis? Do you have any idea about electrophoresis? Electrophoresis means that's a technique which is used in labs. And with this electrophoresis, what and all we can separate? We can do RNA separation, we can do DNA, RNA, everything we can separate, or protein. This and all based on the size of that fragment, or DNA or RNA fragment, or size of that molecule, and based on electric charge. Okay. So for this, electric current is used. It is passing through that gel or the buffer. One buffer is also there. I'll explain you what we are going to do. Here in the first picture, here an electric current that is used through this gel with the help of a buffer. This electric current will pass. Okay, it will work. So that will move this molecule. See, here we are loading that DNA, and that will move the molecule. It is depending upon the size. The movement is depending upon the size and charge. Okay, so that allows the smaller molecule. Smaller molecule will move very fast, faster than larger molecule. Okay, smaller molecule will move very fast compared to the larger ones. Now, next, what I'm saying is, you have to note down, this is not there in your textbook. So in the first picture, you can see a tray. Here you can see a tray. That is the casting tray. Okay, this casting tray, it is a temperature resistant material. Okay, here heat resistant material. Here nothing will happen. We are going to pour a little hot solution on this plate. Okay, but it is not going to damage the tray. Okay, so that is the casting tray. This is, in this casting tray, we are going to pour that agarose gel. Okay, so for agarose gel electrophoresis, which one is the usually used, like agarose? Agarose is, that is a natural polymer which we are using for this gel electrophoresis, normally to separate the DNA, RNA and all. So in this agarose, which is used for the preparation of gel, and for mixing this agarose, say it is in powder form, this agarose that is in powder form. So we need some kind of buffer to mix it, right? Then only we can make it in a gel form. So we need a buffer solution. So first, this buffer we need to prepare. So usually what we will do while preparing this buffer, that will be in a concentrated form. Okay. TB buffer or TAE buffer, like tris borate EDTA, that is the normal buffer which we are using. Okay, TBE buffer. So this buffer, while preparing, what we will do, we'll prepare that in a concentrated form. That means 10x or 50x. When we need, we'll dilute it and use. Usually 1x is used for the electrophoresis purpose. So that time we can dilute it and use. So we used to prepare that in a concentrated form and keep. So from this concentrated buffer, at the time of gel preparation, what we will do, we'll dilute and we will use it. Got it? Then after dilution, now buffer is ready. We are preparing that in a concentrated form. And when we are going to do the agarose gel electrophoresis, what we will do, we'll dilute it. So buffer is ready now. Now we need to add the agarose to that buffer. So first, based on the DNA, we need to find out the concentration of gel. Okay, which molecule we want to separate, or which one we are going to separate, DNA, RNA or protein, whatever it may be. Based on that, based on which one we are going to separate, based on that we are deciding the concentration of gel. Okay, simply we can give any concentration because that depends, that is affecting the movement of that molecule. So what we need to do, we need to decide first which concentration we need to prepare or what is the percentage of that gel. Okay, so while adding that agarose, before that itself we need to calculate the percentage of that gel. Then based on that first step, we need to, which is the first step? First, we are deciding the percentage of gel, and based on that step, we are weighing the agarose. We need to weigh. I told already that agarose, that is in a powder form. So that agarose we need to weigh and take. Okay, the weighing of agarose, that is the second step. Then we are adding this into a flask or beaker, which is having that buffer. We are adding that powder agarose into that buffer. So first we need to decide the concentration or the percentage of that gel. It is depending upon the molecule which we want to get it, which we want to separate. So depending upon that, we are deciding the percentage of gel. Then we are weighing and taking that gel. Right, 2% gel or 3% gel means 100 ml of buffer. How you will make 2% gel? In 100 ml of buffer, 2 grams. Okay, so like that, based on that you will weigh and take. Then you are adding that agarose powder into flask or beaker which is having the buffer. Okay, we are diluting and taking the buffer. Into that we are adding agarose. Then we need to heat. We need to heat the flask or beaker in a microwave oven. So in a microwave oven, we are going to heat this flask or beaker which is having the buffer along with the agarose. Okay, this is to mix. Simply we are mixing means agarose won't get mixed. We need to boil the solution. So we need to get it mixed. So how we will boil? We will boil the solution and we will mix well until the agarose get dissolved. That getting dissolved. Up to that we need to mix it. Then after that, ethidium bromide. What is that? Ethidium bromide, in short form ETBR. We used to write ETBR. What is ETBR? Ethidium bromide. This is to visualize DNA. Okay, here you can see the DNA. Right? Here you can see the bands of DNA. How we can see or how it is becoming visible? By adding this ethidium bromide. Okay. So first, this step we are doing along with the boiling process. That's why I'm saying first we need to boil the solution. Then we need to mix it well until it is getting dissolved. Then after making it cool, we are adding ethidium bromide or ETBR into that mix. Buffer plus agarose mix now we have in beaker. We should make it little bit cool. Okay. While cooling, it will become or it will get solidified. So we should not wait. Almost it is okay means we need to add ethidium bromide to that. Okay. Then this is the easy method which we are doing. Actual process, staining process is later. Okay. So what we need to do here, now boiling over. Now in the second picture you can see pouring the gel. So it is almost cool means that solution when it is coming to the normal temperature, then pour that gel to this casting tray. Now casting tray we need to make it ready and keep. After that we need to weigh this agarose, then mix it with the buffer solution, then boil it and bring it to the normal or room temperature. Then after that, after cooling that, we need to pour that into the casting tray. Okay, this is the normal procedure we used to follow in research and development or during research work in R&D lab. We used to follow the normal procedure. Now after pouring, so after pouring this gel, that will start solidifying. Okay, when the temperature is getting down, that solution, it will get solidified. Okay, so this is the common thing. After pouring, that gel will get solidified and it is forming it will become a gel form. So before getting solidified, in the third picture here you can see a comb-like structure which you have placed. We'll call it as comb. So this comb we need to place. If it is becoming completely solidified, we can't place the comb, right? The gel will break. So before getting solidified, we need to keep the comb. There will be 2 cuts to insert this comb. So it will place in a way that it will make wells. Here you can see the small wells. So to make the wells, we are placing the combs. So this comb was pushed into the gel. We need to push this comb into the gel to form the wells. Here you can see the wells. Okay. Then keeping it like that, this casting tray along with comb and this agarose gel, we need to keep it and later it will get solidified. It will take some time. Okay, it will get solidified. Then after solidification, we can remove this comb. We can take it by holding these both ends. We can pull it and take it. Okay. So after solidification, this comb we can remove. Then if the comb is removed, then you can see wells. You can see here wells like this. When the comb is removed, it will form clean and neat wells. Okay, here you can see the square shaped wells. Then next step, here you can see, in this picture you can see that gel casting tray. Along with that gel, it is immersed into a solution in the buffer tank. Here, this is the tank. Electrophoresis, the tank with buffer. So this gel should get immersed so that when we are applying electric current and loading the DNA, that molecule will move. Okay. So here you should remember that DNA is loaded in the negative side. Here you can see it is loaded in the negative side. Or you have to load because DNA is negatively charged. That's why I explained DNA is negatively charged. So when we are applying electric field, it will move towards the positive charge. So where you have to load the DNA? In the negative side. Because when you are applying the electric field from the DNA should be loaded near the cathode, which is negatively charged. Then same charge will repel, it will move towards the anode. Right? DNA is also negative charge. Cathode is also negatively charged. So when the power supply is switched on, after loading that will move, that negatively charged DNA will move. It will get repelled from the cathode and it will move towards positively charged anode. Right? So first, you are making the glass plate ready, then weighing, first fixing the percentage of gel, then weighing and pouring that into the diluted buffer, then boiling. Next step is boiling the buffer solution with the agarose to make gel. Then after that, pouring that into the casting tray. Okay, then inserting the comb. Next, here comb inserting over. Then loading the DNA in a negatively charged side or towards the cathode. You are loading the DNA and it is moving towards anode. Near the cathode, you are loading the DNA because it is negatively charged. Then we are switching on the power supply. When you are switching on that, the negatively charged DNA that will repel and it will move towards positively charged anode. Got it? Next is, which is the next step? See, here you can see the separated DNA fragments that will be visible only after staining process. Okay. See, when we are switching on that power supply, it will move. But You cannot see that. Okay. So the separated DNA fragments that will be visible only after staining process. Okay. Staining after loading and electrophoresis, staining is the next step. But usually in labs, for saving time, what you will do while preparing the gel itself, that staining compound will be added. Okay, the staining compound will be added. That is just for saving time. The procedure, when we are saying about the stepwise procedure, first preparing the gel, then loading, then electrophoresis, then comes the staining step. Okay. That staining compound, just for time saving purpose, we are adding the staining compound when we are preparing the gel. Next, which one we are adding for staining purpose? Ethidium bromide. So ethidium bromide is the staining compound that is used for DNA staining. Then after staining, when we are keeping this under UV light, these bands will be visible. When the gel is getting exposed under UV light, then the separated bands can be seen. You can see the bands, different bands you can see. Okay, see here. These bands are here. But some other bands, it came little bit down. I said smaller bands or which is having the band size. The size of the molecule is less means it will move. The distance, it is depending upon the size of that molecule. So that's how we are separating the DNA fragments. Okay, here that separated bands that we can cut and we can extract them from gel. Now it is in gel. So the separated bands we can cut and we can extract those bands from gel. That is called as elution. What is elution? The separated bands after electrophoresis, the separated bands can be cut and that can be extracted from that gel. We can extract that bands from the gel. That procedure is called as elution. Okay. So like this, the DNA fragments are separated to make the recombinant DNA or rDNA, and then it is joined with the cloning vector. See, while saying, we are saying like first we need to find out the foreign DNA, then join with the vector DNA, then place in the host organism. It's very easy to say, but these many steps are there. Okay, first DNA electrophoresis we need to do, then we need to separate the band. We need to cut the band and we need to extract them from the gel. Then what is that called? It is called as elution. Okay, extracting, cutting and separating that or extracting that from the gel. That is called as elution. So all these steps we need to follow to separate and make a recombinant DNA. And after this, after separating only, we can join with the cloning vector. So this is how DNA fragments are getting separated. Clear? Next we are going to learn about cloning vectors. Okay, cloning vectors. This topic is important for board exam and NEET exam. These definitions and all that will come for board exam, definition and the properties and all, it will come for board exam. And for NEET exam, you will get application type questions from this. Okay. So what is a cloning vector? That is the first question. What is a cloning vector? What is a vector? So cloning vector, what do you mean by cloning vector or what is that? Cloning vector means that is a small piece of DNA. That is also DNA. That is a small piece of DNA. So this DNA, that foreign DNA or the DNA fragment which is having the gene of interest can be inserted for cloning purpose. Okay. Cloning vector, what is that? Cloning vector is also a small piece of DNA. That is also DNA. Otherwise, you cannot stick the DNA which is having the gene of interest with the vector. Both are DNA fragments only. That is also a DNA, a small piece of DNA. To this DNA, you are going to join the DNA fragment which is having the gene of interest. Okay, so both will join. Then we are moving to a host organism. Got it? So cloning vector is a small piece of DNA, and to this DNA, that foreign DNA or the DNA with the gene of interest or the DNA fragment with the gene of interest can be inserted for cloning purpose. So vector DNA is a DNA molecule and that can carry the foreign DNA into host DNA. Without vector, it is not possible to carry the DNA molecule into the host cell. So with the help of this vector DNA, the DNA molecule can be easily carried to host cell. Okay. So another property of vector, that is this vector can replicate, or vector is having the capacity of self-replication. Vector is having the ability or capacity to replicate. Okay, replication, that is having the capacity of self-replication. So vector So what and all vector can do? Vector can carry the DNA molecule into the host cell, the DNA with the gene of interest or the DNA fragment with the gene of interest. A vector can carry that into the host cell. Then another property is that is having the ability to replicate, or vector can self-replicate. The next property, vector can integrate into host cell. Okay, both will be having similar type of sticky ends with the help of restriction enzymes that will make same type of sticky ends so that the gene of interest, the DNA fragment with gene of interest can join with the vector. Okay, so both can join and they can move to host cell. Bacteriophage, plasmid, these are all the examples of vectors. Examples for vectors are plasmid, bacteriophage, then TI plasmid. Hope you have learned in the case of tissue culture and some tissue culture work we are using TI plasmid. So these plasmid or bacteriophage, they have the ability to replicate within the bacterial cell and there won't be any control of this chromosomal DNA. Okay. So just now I said this bacteriophage, they can multiply without any control of the chromosomal DNA. Okay, this bacteriophage is having the capacity of self-replication, or this vector is having the capacity of self-replication without the control of the chromosomal DNA. So bacteriophage that have high number per cell. Okay, that means in a bacterial cell, what do you mean by that? In a bacterial cell, number of bacteriophages that can be more, and along with that genome, this copy number also increases. Okay, I said there is no control of the chromosomal DNA, right? See, that chromosomal DNA won't be having any control in this vector. So that vector will multiply along with the copy number. Here this also can, along with that genome, that bacterial genome, this copy number, that is also increasing. So that's why these are selected for cloning, because we'll get more numbers. In a faster way, we'll get more numbers. That's why we are selecting bacteriophages as cloning vectors. Bacteriophages, these are the examples for cloning vectors. Plasmids, bacteriophage, TI plasmid, all these are examples. So why we are selecting? There is a reason for selection. What is the reason? There is no control of the chromosomal DNA. So in a bacterial cell, number of bacteriophages can be more, and along with that genome, this copy number is also increasing. So that's why it is selected as a cloning vector, because we need more clones with that gene of interest. We need the gene of interest along with more number of, we need more numbers. That copy number should be more. So with that gene of interest, we can create more copies within a restricted time period, right? Within a limited period, we can make more numbers. So in the case of plasmids, now we were discussing about bacteriophage, right? Next, in the case of plasmids, some plasmids, they have the capacity of 1 or 2 copies per cell. Okay, the plasmids, some of them, they have the capacity of 1 or 2 copy numbers per cell. But some others, they have the capacity of 50 into 100 copy numbers. And more than this also there. But some are having 1 or 2 copy numbers per cell. They have the capacity to have 1 or 2 copy numbers. But some 50 to 100 copies. See, suppose we are making a recombinant DNA. For that, we are linking a foreign DNA or a piece of alien DNA. That means the DNA with the gene of interest. Don't get confused with a plasmid or bacteriophage, which is a vector. Okay. Then, along with that, the multiplication of this plasmid or bacteriophage, our linked foreign DNA, that is also getting multiplied. And we are getting many copies of that. So how that process is happening? We are making a recombinant DNA. So what we are doing there? We are linking the foreign DNA or the DNA with the gene of interest to a vector DNA. That vector may be plasmid or bacteriophage or whatever may be. That will work as a vector. Then while joining the foreign DNA with the vector DNA, along with that, the multiplication of that plasmid, the plasmid is getting multiplied. Right. So along with that, our foreign DNA, that is also getting multiplied because we have linked both. Then we are getting many copies of that gene of interest. Right. So now, with the help of this biotechnological processes, scientists are designing vectors. They are designing many type of vectors. So the vectors are designed in such a way that the linking process and the selection of recombinants are easy. They are making it very easy. They are making vectors in such a way that the selection process and the linking process. First, we need to link the foreign DNA with this vector DNA. That should be easy. Then only they will select the particular vector. And selection process of recombinants. We should be able to select the recombinants. See, after multiplication, we'll get so many copies of that, right? From that, we should be able to select very easily which is the recombinant one and which is non-recombinant, which is having that foreign DNA part and which is not having. We can't select all. If 100 copies are there, means we can't select 100 copies. We need to select them, select the recombinants from them, which is having the gene of interest. Right? For that only we are trying. So we need to select the recombinants very easily. So based on that, nowadays scientists are designing the vectors. So the plasmids and the bacteriophages, they have the ability or they have the capacity to replicate within the bacterial cell. And they will be independent of the control of the chromosomal DNA. Okay. And why we are selecting bacteriophages? Because they have high number per cell and they have high copy numbers of their genome within the bacterial cell. And some plasmids, they may have 1 or 2 copies per cell. But some other type of plasmids, they may have 15 to 100 copies per cell. Okay. So how we are designing, or nowadays how we are designing the vectors? Based on what and all? For linking the foreign DNA very easily, and the selection of recombinants, that also should be very easy. So keeping these two, keeping these two in mind, we are designing, or the scientists are designing new vectors. Okay. Next, there should be some special features while designing or while selecting a cloning vector. So we should have some minimum features. Which are they? First one, origin of replication. There should be origin of replication, then selectable marker, then cloning site, then vectors for cloning genes in plants and animals. So origin of replication, selectable marker, cloning site should be there, then vectors for cloning genes in plants and animals. So these are the features which is required to facilitate cloning into a vector. So a cloning vector, that should have an origin of replication so that the vector can self-replicate inside the host cell. Okay, why a cloning vector should have an origin of replication? If a cloning vector, that should have an origin of replication so that that vector can self-replicate inside the host cell. Okay, for inserting the foreign DNA, a vector should have a restriction site. We have learned how restriction sites or restriction enzyme, how it is working, and how we are making sticky ends, and how we are joining the foreign DNA with the vector DNA. We have already learned that. So that is the, for inserting the foreign DNA, that vector should have a restriction site also. Then only that restriction enzyme can cut and make the sticky ends for the insertion of foreign DNA into that vector DNA, right? Then that cloning vector should have a selectable marker. Cloning vector, that should have a selectable marker. Usually antibiotic resistant genes which we are using as a selectable marker. Antibiotic resistant gene. Okay. Those antibiotic resistant genes, those genes we are using as selectable markers. That means the marker which is helping for the artificial selection. Usually for what purpose we are using markers? For the selection process. Okay, to mark something. That is marking something, or that is helping for the selection. For artificial selection, it is helping. So the antibiotic resistant ones, they are the recombinants. The recombinant ones will be having antibiotic resistant capacity, or they have the capacity to resist. Okay, so the antibiotic resistant genes, they are used as the selectable markers. Antibiotic resistant ones are the recombinants. So the selections can be made very easily when we are using antibiotic resistant genes as the selectable marker that we need to study in detail. Then that vector should have multiple cloning sites. That is another feature. That should have multiple cloning sites. That is the next feature. So that should be suitable for working with prokaryotic and eukaryotic system. This vector should be suitable for, that's why we said the fourth point, vector for cloning genes in plants and animals. So that should have or capacity, or that should be suitable for working with the prokaryotic and eukaryotic system. For both systems, it should be suitable. And the vector, that should be small in size, so that it can be easily integrated into the host cell. Okay, so that vector, that should have the capacity to accommodate larger DNA Means that vector that should be having or that should be suitable for accommodating the larger DNA segments also. Okay, so like this, vector should possess many qualities, and we need to study all these points in detail. The origin of replication, what is the importance of origin of replication, selectable marker, cloning site, then vector for cloning genes in plants and animals. All these points we need to discuss in detail. In that, selectable markers. What is the role of selectable markers? That I'll explain you, and which are all the common selectable markers which we are using, and what is the role of origin of replication. Then how we are selecting the recombinants and how we are discarding the non-recombinants. All these we need to study in detail, and these topics, mainly these topics, from this chapter you'll get a lot of application type questions. So go through all these things. And if you have any doubt, you can ask me in the next class. Do you have any doubt? Okay, hope everything is clear. Just go through this portion. If you have any doubt, you can ask me in the next class. Okay, thank you.